activation of the genes that are used to produce induced pluripotent stem cells. Many of you know that Francis Collins, who was from the University of Michigan, ran the federal search to map the entire human genome. The way that was done was samples were taken from several different human beings and combined so that we could get a map of the human genome. And it's the same kind of approach that is used for the samples that you're talking about. That part, Senator Berger, has not been controversial. The controversy has really focused around destruction of an embryo to create new embryonic stem cell lines. Other questions? Yes, ma'am.
I was wondering if stem cell research is or will be useful in the study of mental illness like schizophrenia.
That's hard to say.
Just
Yeah, will stem cell research be useful in the treatment of mental diseases such as schizophrenia? At this point in time, it seems unlikely, although one can never predict where it would go. Those diseases are typically multi-genetic and have more than one gene at fault. And the diseases which are particularly amenable for stem cell therapy are those due to the loss of a particular cell type in a particular location. In a disease like that encompasses so many areas of the brain, it seems unlikely to imagine how stem cell therapy might help at this point in time.
You mentioned the problems about using induced pluripotent stem cells. What are the drawbacks of using embryonic stem cells? What are some problems you're facing?
Well, one of the drawbacks, the question was, you've mentioned the drawbacks of iPS cells. What are the drawbacks of embryonic stem cells? I would say there are two at the forefront. One is the ethical controversy associated with them. Not everybody believes that that's a way forward to treat and cure disease. The second is a problem of the immune response of the body, such that like any organ transplant we now do, that organ or tissue has to be matched at a certain level between the donor and the recipient, otherwise it's subject to the immune system rejecting it in the recipient. That would be the second issue. The extent to which the cells derived from embryonic stem cells elicit an immune response, I think is still up in the air. That particular trial from Geron, where they're trying to treat patients suffering from spinal cord injury, in the information they provide on their website, which is limited, they do say that they only expect to administer a short-term course of immune suppression for this particular therapy. And in basic science studies published, using this cell line in an assay to kind of gauge the response of the immune system, it's relatively benign. That's in an artificial system. It's yet to be shown how that will work in humans. But those are the two aspects, the two biggest, in my opinion, drawbacks at this point in time to embryonic stem cells.
questions on this side? Yes, ma'am.
How long are the stem cells viable?
The question was, how long are the stem cells viable? So the stem cells themselves, once we have them in a laboratory setting, are viable for a long time, in some cases years. It depends how you care for them, I guess. And there are ways in the laboratory in which we can always kind of have a fresh pot of cells that we haven't manipulated very much. So they have a very long lifespan in the laboratory.
So there is, just to give you some scale, one of the most famous cell lines created, not a embryonic stem cell line, but just a line created from human cells, in this case from cancer cells was created at Johns Hopkins in the 1950s and is still viable today.
With respect to those cell lines, do you have like a master list of like who's got what cell lines, and you might contact Georgetown University for some you need, they have some special kind, or USC or something, and by the same token, or you have to keep track of them, you have to keep track of your stem cells like you would drugs in hospital settings, you know what I'm saying?
I think I do.
You're
So,
you're not selling them on the black market.
so it was a two-part question. One, is there a repository where we get these cells, or a central database where you can see who has what type of cell? And the second part was, do you have to keep track of them? So the first part is there are multiple databases of who has what stem cell. There's an international database kept by the International Stem Cell Initiative. There's a national database kept at UMass Amherst, I believe is the campus, and other ones around. So you have to kind of cobble together between those to know what's out there, but that's still likely an underestimate of what's out there. Those are only the ones that people have registered and made available. When we then get the lines in the laboratory, we do have to keep track of them. And maybe this is a little too technical, but there, when President Obama changed the funding guidelines, the NIH still hasn't determined which lines will fit under that guidelines and which ones won't. So we have kind of two separate pots of cells, one that are NIH approved and those that are yet unapproved. And it's those unapproved those lines that we have to especially keep track of and be careful that we don't use any federal money to study them or experiment with them.
And it is that September 21st article that I referred to that talks about NIH creating a group of scientists led by Jeffrey Botkin at Utah to characterize those lines and decide which of those can be used for federally funded research. There will be a central government federal repository that will keep track. What we do at Michigan under our escrow committee is keep track of what every researcher is doing, what lines they are using, who is using them, and what are they being used for. So is it possible to have a black market? It's always possible to have a black market. It would be very difficult to have a black market of cells in the United States. Way in the back, yes ma'am.
What lines are for the fountain of youth or faster cell refresh?
Sorry, I didn't hear the question.
What lines are for the fountain of youth faster
Yeah,
cell
so the question is, actually, and it sounds facetious, but it really isn't. The question are, what lines are for the fountain of youth? Jack and I actually saw an article that said that there were some people studying cell lines to reverse the aging process. So it's not as far-fetched as you might expect. Because if you can figure out why cells age and how they age and what things like stem cells can do to retard or reverse that process, you're on the track of your question. This is, the work that is being done with stem cells is among the most exciting work that is being done in science today because of the almost unlimited potential. So those are not questions out of the bounds of asking, although they do have profound ethical implications. In the front, please.
I understand that legitimate researchers attached especially to research universities are going to be following all the scientific and ethical guidelines that are out there. My question is for those people who maybe are not attached to such reputable research institutions, is there any possible foreseeable problems from since you guys really understand the leeways of the language of Prop 2, do you see any possibilities where there's some, because it wasn't super scientifically precise, where maybe some not quite so ethically ethical researchers could have wiggle room to be doing research